get started. First off, I'd like to thank everyone for joining, and particularly, of course, Neil, I'd like to thank you for taking the time out of your day to join us virtually, at least, and present some of the, I guess, some of the work that you've been working on here, building a community of top-down and native proteomics. So Professor Neil Kelleher joins us from Northwestern University, where he is the Walter and Mary E. Glass Professor of Molecular Biosciences. He's also the Professor of Chemistry and Medicine and the Director of the Proteomics Center of Excellence. Neil is a transdisciplinary investigator who is making an impact in the field of proteomics, proteins, and the discovery of new antibiotics and anti-cancer molecules. He runs the Kelleher Research Group, which invents methods of understanding how human cells work at the molecular level. He also runs the Northwestern Proteomics, the leading lab in the world for top-down proteomics approach to measuring proteins. In 2016, he delivered a TED Talk about cell-based human proteome project, and this is an effort to map 1 billion proteins throughout the human body. He's also the founding president of a worldwide research consortium aiming to actualize this project. He has quite a long list of research contributions that are listed here below, but without further ado, I'm going to turn the ball over to Neil, and he's going to start the seminar. So Neil, I appreciate it. I'm going to stop sharing my screen, and hopefully you'll have the capability to share yours. Yes.
That's right, yeah. Thank you, Christopher, and everybody there in the Delaware Valley region, and the great to get a chance to come back and virtually. Let's see if I can get this into presentation mode. And when I did that, can you still see the title slide?
Yes, we can.
Aha.
Looks great.
Okay. Well, brilliant. You know, thanks for taking time out of your day and to go into Zoom land with me and talk about my favorite topic, and that's whole proteoforms, whole protein molecules, and their readout by mass spectrometry, which now includes their complexes and biomolecular assemblies by native proteomics. And the community building aspect is something as I progress and get ever more gray hairs, think about trying to bring together us and do something very significant before we're all pass on to next generations. And so for me, and Christopher mentioned it in the outset about 1 billion proteoforms for $1 each. That's a soundbite or a tagline, like an elevator tagline, that I'm increasingly voicing with leadership at NIH and other national institutes, federal funding agencies. That would be 1 billion redundant proteoforms. I don't believe there's that many to really break open the world of proteoforms and to, in effect, sequence the human proteome. So that's a giant mouthful. And we are going to tangentially cross that about mid-talk. And I also invite chat box and raising of hands, etc., throughout. A two-way street is much more interesting than a one-way diatribe. So that's a super provocative start. And I can imagine people's responses, maybe particularly in the private sector, when we think about, you know, framing the human proteome and what it would be about. So that's a different kind of talk, and that one's maybe saved for another day. What I thought I would do instead was to give you an update about some very granular aspects, community building, a way to be less provocative about what I just said and to be able to, but still move on it as we, you know, open up to this decade, which involves not just COVID-19 and SARS-CoV-2. And I'll say just at the outset too, before I dig into the details here, that I've become a budding virologist and immunologist, and many of you undoubtedly have gotten pulled into this vortex of focusing on the virus and its effect on our society. For our take, we're getting very interested in IgGs and serological testing and how we might improve that. And some of the technologies I'll talk about today, I think can do that. So we've been very active in that in the past few weeks. And there's a podcast called TWIV, This Week in Virology, and it's someone in your general area there, the Eastern Seaboard. His name's Vincent Raccagnolo. He's at Columbia, and his podcast has just exploded. It's called TWIV, and I encourage you to, I mean, they do long form interviews, basic science, translation. It's just spectacular what you can learn. I've just been chain smoking those TWIVs. Okay, so let me just sink into the details here, and please stop me. I'm going to be sort of a freight train moving forward, but please stop me if I can clarify, or I don't need to get through all 186 slides. Okay. And yes, that's a joke. What I'm going to do is get you just really brief updates on top-down proteomics, what's changed in the last few years. There's, of course, two modes, denatured and native. And denatured is just as it sounds. These are mainly analytical words. So native is in quotes for a reason. I suggest we don't have that debate about gas phase and solution phase. I'm mainly coming at you with compositional analysis, the atom composition of the protein molecules. That is our power stroke, and I'll describe more about that. But we'll be oscillating between these two modes of analysis. And so just as we've talked for a long time, whether it's chromatin biology and histones and their composition, like Ben Garcia does and his group at Penn, or complexes, biomolecular assemblies, we can now apply mass spectrometry. The last 5 years has just, it's been a veritable explosion in commercial grade technologies to do what I'm talking about. And that's very different than 10 years ago. So we'll apply this, this value, and the value proposition is something I'll try to sneak into my talk, about getting the full composition of protein molecules and not through bottom-up methods, just pieces. And I'll end with a really, I think it's going to be a big breakthrough. History should read that out, of using individual ions and single particle mass spec, something we've together with Thermo broke open in the past year or so. And it basically, the soundbite on this one takeaway, and I'm a believer in talks about bluffing people, bottom line up front, bluff. And basically this bottom bullet here, the individual ion MS, it now allows us to tackle any target in biology, and we can get a mass readout. Like this virus-like particle here, formerly you would be in the mass to charge domain and you couldn't get a mass. You wouldn't resolve the charge states. You'd be, there's no information there. But now we can put a mass scale directly on that by tracking the exact charge on each and every ion. Okay, so that's what I'm going to talk to you about today. And I've already given you the big preamble, which is atom composition. We've now had this well-articulated trichotomy of different analysis levels, and they each have their unit of currency, like the tryptic peptide being bottom-up proteomics, and of course, so many labs do that, and that's a approaching $4 billion a year ecosystem in mass spectrometry-based proteomics to make tryptic peptides and bottom-up and in general flow nicely. So it's a very high value, well developed, and we're continuing to develop it as a community. So what I'm envisioning is another ecosystem about that large for these other two levels of analysis. So one is the denatured level, the level of the proteoform being the unit of currency, not the smaller little tryptic peptides, but the entire proteoform. And then the same articulation, the same value. Don't infer your way to a level up on this tri-level diagram. Directly measure it, directly read it out. And that has a whole separate conversation too about protein inference and direct measurement. I'm going to also sidestep that. There's 6 points under that explain the really nuanced view of top-down and bottom-up. But I'm going to skip through that pretty quickly. But the general value proposition is that when you pain yourself to go up this tri-level diagram, to go at the higher levels, you'll be rewarded. And what does that mean? Well, the things that you measure, the molecular entities that you measure, and the composition that you assert per measurement will be of higher value. So if you want to get combinations of post-PTMs, or you want to get polymorphisms as they occur in combination with isoforms or PTMs, it's the variation in all of their combinations that you get out. That's the general statement. And when you do that, that they should connect more tightly. And that could be true in basic research, fundamental biology about putting function to PTMs, post-translational modifications, assigning function to them and detecting them reliably. There's a value proposition there. And then there's also one in biomarkers, and to do clinical research, but have it be proteoform resolved. So in this middle level of tier of operations. And the rock curves that you get out, the correlations to complex phenotypes would be stronger. And we're in the process of making that data-driven, not a hypothesis. So that's the general layout as atom composition for proteins hits this 3 different ways to read them out by mass spectrometry. Here's these 2 modes, and I'm going to start to pick up some steam here. The 2 different modes, like here's the same molecule, it's enolase, denaturing mode on the left, all the different charge states in a Q-exactive style mass spectrometer. And on the right, it's the same benchtop instrument, it's just tuned for native mode. It's a UHMR style instrument, and we're at pH 7. You get far fewer charge states, you retain cofactors like metals, and if there's a complex, it will stay intact, more or less. And these are the 2 different worlds. And of course, in chromatography, you're going to get the left-hand world for denatured pH 3. And in native world, we'll come to how you might solve the separation problem and how to do native top-down, like you can do denatured top-down, except at much higher mass with fewer charge states and all sorts of other advantages. So, and now there's commercial platforms to do both. But let's get denatured first. And so here's the proteoform. If you're not familiar with this, there's a 501c3 based in Boston. It's the Consortium for Top-Down. And we're in the process of strategic planning, and we should have a white paper out soon this year about the future of top-down, et cetera. And in 2012, when we formed this group, and thanks to the vendors really allowed us to form a group and to be more precise about proteoforms and what they are. And it's like what I said, right? It's the combination of modifications and all the sources of variation. So just quickly on the right-hand side, what we've got here shown is like a phosphorylation, the pink circle, a glycosylation, a particular isoform, an exon region is displayed here, and a polymorphism, like a coding single nucleotide polymorphism. So all those combined, right? That's the proteoform. And that's what we can measure if you don't digest into peptides. So, it's reasonably well cited. And now we're in the process of tying proteoforms to biology. And that's basically what I do for a living. So in 2018, Lloyd Smith and I tried to not provide this message inward looking to our field, but go outward looking and to genomicists and geneticists. And so in a one page, one figure paper, we tried to make this case and tried to highlight with nuance what it means to infer from peptides into what from a single gene you could get an entire proteoform family, related proteoforms from the same gene. And then, well, I'll leave you to peruse that one page article at your leisure. The consortium's done a lot of things. It's published 7 different articles over the years, and here's the latest one in Nature Methods. It's a more precise way to classify different quality levels of proteoforms. And so there's these 5 different quality levels. Like a level 1 proteoform is just, perfect. You know where the PTMs are, you know that the sequence is defined, and you're absolutely sure which of the 20,300 human genes, if it's a human proteoform, gave rise to the proteoform. So there's more reading there, and you know, we're trying to then now map scores and algorithms and software into this 5-level quality system for proteoforms. Yeah, so, but let's move into native mode. So you've probably seen a lot about denatured top-down if you're in the field. And so let's switch into native mode. And here, big things can fly. So, you know, complexes 10, 20 megadalton have been, and even higher, have been detected using native mode, native MS. And I don't know how far we'll get in size, et cetera. Here's a picture that Ole Jensen gave me. Now, this is an intact organism. This is a mosquito that got stuck in the inlet to his mass spectrometer. And I don't know what the molecular weight of a mosquito is, but I don't think we'll quite get that large. Okay, moving on. That's kind of dry by Zoom, right? You know, you just, it's kind of hard to hear people laughing on mute. Anyway, my point is we're getting higher in molecular weight, and why do this, right? I kind of described it at the outset, right? If you compare what biology produces in the center, like here's a complex, it's an alpha-2 beta-1 trimer, and it's got a zinc bound because that's the metal that's biologically and functionally important. It's got a substrate that could be covalently bound, and then a glutathione is attached to a surface cysteine, right? If that's our answer, and then on the left-hand side depicts bottom-up and what the information you get, and on the right-hand side is what native proteomics or native top-down will give you. Well, this is a picture of trying to be compelling about why you would adapt what you do in protein mass spectrometry to try to read out more directly the composition of complexes and the stoichiometry of the subunits. Okay, so we're picking up speed here. So this is deriving from something that lots of other labs, not us, not my lab, years before, people had known that if you take a homotetramer like pyruvate kinase and eject it and heat it up, right, after native mass spectrometry, you eject a monomer. And when you do that, it supercharges. It asymmetrically steals a whole bunch of the protons when it leaves the parent complex in the gas phase. And it's thought that this subunit unfolds and will steal a bunch of protons as it unfolds. And that's why it gets almost 3-fold lower in mass to charge for the subunit versus the parent complex, because it, 2,000 relative to 6,000. It's because the Z went way up relative to the M. And that was a gas phase phenomenon. So that was known. So we applied to the Keck Foundation, got $2 million to then do this experiment, to put the down on the subunits. So this is what we call native top-down. And then if you don't know the identity of the complex that you're starting with and doing this in discovery mode, well, that's native proteomics. Moving right along, we had worked with Thermo starting in 2011 to adapt the Q-Exactive platform to go out higher in mass and do it native. In late 2018, this was released commercially as the UHMR, and it now goes out routinely to 30,000 mass to charge or even higher. Won't get into all the details of that. It was published in this paper here in 2013, where we first showed, take a controlled disassembly of a protein complex. So I'll go through that. This is the team that did it back in the day, including our Brazilian group here. Luis, Henrique, and Rafa have all been great in the lab. And Henrique will graduate soon, within the year or so. Right. So our instrument, this is basically that experiment. There's a tetramer, and I'm not sure how animations do on Zoom, so I'll tread carefully here. But there it is. There's a tetramer. We don't know the identity of what it is, and we're going to read it out by mass to charge. There it is, intact charge states. And then we can isolate. We can actually use the quad at this high mass to charge range, isolate a given charge state, and then eject in the HCD cell. Hopefully you were able to see that. Eject that monomer and do that supercharging, which you can again see evidence for that in this spectrum featured in this animation. Okay, and I'm going to come clean here. We're not doing MS to the third. We sort of cheat a little bit. We call it quasi MS3 or pseudo MS3. But here's how we get and do top down on this subunit from a homotetramer. Okay, so we want to isolate that 16 plus. What we do, and I know this is being recorded for students in a course, which is awesome. So you can have a quiz on this part of it. We eject in the source the monomer. So we apply the vibrational energy in the source rather than the HCD cell, because we need the HCD cell to do the fragmentation of the monomer. And you know, it's our belief that you could build a mass spectrometer that was better tailored to do true MS to the third, but we sort of sneak it, and it's carried us a long way over the last few years. But so to be clear, we eject the monomer in the source, and then we isolate in the 16 plus of that monomer, and then we fragment it, and now isolated. So formally, the connectivity of which monomer goes with which complex, it can be asserted with confidence. It's just I wanted to come clean about that. Okay, but now we've got backbone fragments. Now we can put it into a search engine and identify which complex it is. And so I'll take you through one of those as I move through relatively quickly. So native proteomics, if you imagine at that three-level diagram at the top level, right? Could you ever imagine one day doing native proteomics, having separations, extractions on the far left-hand side of the slide from a whole lysate, a whole, you know, the target of proteomics, and then doing what I just showed you, which is the center part of this slide, MS1, MS2, MS3, and then a search engine. And so that's what we had put forward to the Keck Foundation. The WM Keck Foundation loves high-risk stuff, and I mean, this is the kind of thing at the study sections or under peer review that are like, well, that's high risk. We're not into it. There's all sorts of reasons why this wouldn't work. But the Keck Foundation loves this stuff. So, and indeed, after, you know, a few years, we can now do this and show that it's possible. So what we did is take HEC 293 cells, do an ammonium acetate separation on a big fat ion exchange column, like, you know, 4.6 millimeter column. And I'm just going to show you one fraction. So we fractionated an entire cytosolic cell lysate. And then here's one fraction. You know, we did this over like 70 fractions and reported it in Nature Chemical Biology 2018. So here's an example complex. It's just one of about 15 on average. We see 15 different complexes. And what are they going to be, right, students? They're going to be the most abundant small protein complexes in the cell. Primary metabolism, glycolysis, TCA cycle, mitochondria. Those are, in fact, over 70% of what we saw. In this case, we have 89 kilodalton complex. What is it? Well, we do our thing, the thing I just showed you. Eject the monomers. There's the ejected monomers. There's the intact tetramer left over. Then we fragment, we weigh the intact and the monomer, and that's 22 kilodalton monomer. So the intact mass is around 89 kilodalton. So you do this clear step and say, well, it looks kind of like a tetramer, 4 times the mass. It's a little off. It's about 200 plus daltons off, and we'll get to that. And then we do the subunit fragmentation, just like I showed. Okay, and all of these little fragment ions down around the baseline, we then input those into a search engine. We isotopically resolve them all. You do have to be cognizant of the pressures, the pressure in the HCD cell and how much pressure you leak into the Orbitrap. You have to play around with that a little bit. It's called protein mode. And it's different if you have a whole complex, big complex, or these smaller fragment ions. But nonetheless, I digress. We take the fragment ions, we shove them into a search engine like ProCite, And here we can identify the complex. There's also a database of complexes, so human protein complexes. There's something called Quorum, and all of that's been loaded into a search engine that's web accessible. And so either one of these search engines comes up with this result. So all those fragment ions led us to be extremely clear that 1 × 10⁻⁸ raw p-score for this hit. And it's human SOD2. And not only that, it's not SOD1 or SOD3. It's definitely the mitochondrial version, SOD2. And there's our information locking down both the N and the C terminus with all of these B and Y fragment ions mapped to the primary sequence during the search. Let's get back to the reason it has this mass difference. So that's a subunit mass difference. Oh, I see I'm missing a slide. Yes, I'm missing a slide. Okay, the 210 Dalton mass difference, which is what it was between 4 monomers and the intact complex, what that is due to is 4 manganese ions bound, which are for each tetramer, they have a divalent manganese as part of SOD2. That's very different than the other SODs. So you pull this all together and you get, within a Dalton confidence for the entire complex. Sorry, I missed a slide there. So not just that, there's more as we move to close this vignette down. In this complex, SOD2, the tetramer, that's the mass I was showing you here on the far left. And what this was Owen Skinner. Owen's a postdoc now with Vamsi Mutha at Harvard Med, and he'll be on the job market pretty soon. He was a fantastic graduate student. And what he saw was that if he was really gentle on the front end of the source, he would see these adducts at what turns out to be 33 Daltons exactly. This is most consistent with the product, or excuse me, the substrate of SOD2 being bound and up to 4 times. And so the model for this, and we actually have never gone back and really studied it in vitro and shown that we can actually trap this putative superoxide stuck to as a non-covalent substrate bound to the protein. But if you go through the math, 33 Daltons is most consistent with superoxide, not one of the products, hydrogen peroxide or oxygen O2 bound, but rather superoxide. So, that's the power of native MS, is to preserve labile ligands, or substrate in that case. So here's a survey of what we did, 125 complexes. We didn't know which ones were which until we did this native proteomics approach. They're all below molecular weight. I mean, our first instrument really couldn't go beyond 500 kilodaltons in size, and that means about 10,000 mass to charge. 9,000, 10,000 mass to charge is about 500 kilodalton. Based on the way that charging happens in native MS. We found both native proteoforms, so monomeric, but also multimeric, mostly alpha-2, alpha-4, alpha-6 homo-oligomers, or complexes of the same subunit, as what you see in glycolysis and the mitochondrial abundant complexes. So all that was published in 2016. I now want to pivot. We found 28 metalloenzymes, and 3 of those were novel. And new metals were found to be associated with some of these complexes. A lot of cysteine surface modifications could be found, and that's another benefit of native MS. So that's that story, and I'm going to pause, grab a bit beverage here while maybe I allow time for a question or two, but Okay. I know I'm gonna, if that was, I hope the rate is okay. And audio is good. You can hear and see everything okay, Chris?
Yes, we can see and hear you great.
Okay. Okay, perfect. Well, I'll start to accelerate a bit even further, I think, to try to keep or capture attention. One of the things that came out of the native study, like here's a classic alpha-2 case that came out. Alpha-2 dimer from primary metabolism. It's enolase. It's alpha enolase, but endogenous to these HEC293 T cells. Okay, and you go, okay, alpha enolase, great. Okay, how many PTMs are on this thing? How many modifications are on it? And if you go to SwissProt, which aggregates a lot of data from bottom up, you will find that there are 35 post-translational modifications on 27 residues. And it's the kitchen sink of mods. And you go, oh my God. So if you think about what kind of proteoform complexity that would conjure up in one's mind, you would go, oh my God, at least 2 to the 35 proteoforms, which is like some cosmologically large number. When we look at the ejected monomer, to give you a sense of this, so this is a deconvoluted result of the monomer that came out of that complex, we don't see high stoichiometry mods. And so this would be 1 phospho, where this arrow is, 1 malonyl or succinylation, right? They're either very low stoichiometry or in this biological context, not present with abundance. You know, there's an acetylation on the N-terminus and the canonical form. So, you know, I think that has significant implications that we don't observe a lot of these PTMs, at least for small primary metabolism type enzymes. So it gets you thinking about the whole proteome and what is the expected complexity. Now this differs greatly, and let me orient you to this graph here on the right. So this would be the human proteome gene number. There's 1 to 20,300 or so human genes, okay? And then on the y-axis is the number of proteoforms. So I take us up to just 10,000 proteoforms. And so what is the model? Like maybe alpha enolase is something, you know, down here, right? Where it's one of these few thousands of genes that don't have a lot of post-translational complexity. But then something like a favorite of Ben Garcia, Histone H4, might be something on this right-hand side of this, where Histone H4 is decorated with all sorts of high stoichiometry and low stoichiometry combinations of modifications. Ben and I have tossed us around over a malted beverage and, you know, sort of said, well, maybe 5,000, 10,000 proteoforms at some level of depth, like how low you could go in detection limits defines how many proteoforms you would see. So, you know, my estimate is something like 10,000 for Histone H4, which there's different flavors of Histone H4. But that would be a very highly complicated type of gene product or set of gene products. So it just gives you a sense of, at least we've got dimensions X and Y on this graph. And this, I think, is important for the future of a human proteoform project and what we might try to do to try to regularize this area. I think basically the take home is, I don't think it's as complicated as a straightforward combinatorial explosion of all the inventoried PTMs would lead you to think. Okay? So there's Histone H4. And you know, I tried to socialize this idea with about 70 of my close personal friends on this paper. How many human proteoforms are there? So I encourage you, if you wanted to drill down on this further, you know, it's the difference of possible variability on the left. Like you got all of these sources of protein variation, but it's my belief and assertion that it's nowhere near that combinatorial explosion. That for some proteins perhaps, but the rule might be that biology has evolved to relatively constrain the canonical use of PTMs because it is energy bearing, and certainly high stoichiometry logic gates. Now, I'm not saying that environmental exposure, obviously if I breathe in a whole bunch of really bad chemicals, I'm going to be alkylating a lot of my proteins. So environmental low stoichiometry mods are a separate conversation. But it's complicated, so, but at least we can start to put axes on different dimensions and talk in a more regularized way. Okay, and so I'll take you through this one other example, Histone H4. Same deal, if you go to SwissProt, you look up the accession P62805, and you pull up Histone H4, you'll find, at least when we did this, 66 PTMs at 17 different sites, which is well over 1 billion different theoretical proteoforms. Question, how many actually exist? Well, if you do some math, here's some common PTMs. So let's shave it down to 16 most common PTMs at 13 sites. So here's all the different sites of modification. Some lysines can have mono, di, and trimethyl groups on them, and acetyl. So they have up to 5 types of PTMs at a single site, some even more. So, but under, general biochemical constraints that a chromatin biologist would say, okay, how many would you get? In this scenario, you get 98,000 proteoforms for just histone H4. And we've created a little tool where you can do this. It's called Protein Annotator. And you can kind of play around with PTMs and then boom, hit calculate how many proteoforms do I get, et cetera. It also spits out XML sequences that can then be used for searching, et cetera, or mapping fragment ions and top down. That's just the math that governs the PTM explosion, and the number of theoretical proteoforms explodes according to this equation. It is exponential. But histone H4, and this is something the consortium did, and Ben Garcia did years ago. And in this paper, we shipped histone H4 around to 11 different labs. They reported back, and the answer was 75 proteoforms with confidence across about 4 to 5 logs of dynamic range. And now there's more. Histones are very abundant, small proteins, but it gives some sense that there is a constriction to the combinatorial explosion. And Ben's postdoc, Nick Young, who just got an award, US HUPO Young Faculty Award. He's at Baylor in Texas, and he's found now up to about 175. So he's digging deeper down the iceberg and uncovering more and more proteoforms. But back in the day, this arginine 3 dimethyl and arginine 3 monomethyl were sort of right down here at the bottom of the iceberg. And yes, we need better technology to be able to push lower, et cetera. And such would come out of a human proteoform project. Say, for example, after we get rid of this virus and get a vaccine and everybody gets their immune passports and goes back to work. So let me just show you the native mode going higher up that tri-level diagram. So remember the bottom up, and then the proteoform level, and then native top down. So this one is for the chromatin folks out there. So this simple experiment, we just took the native, the whole nucleosome, which is about 200 bases of DNA wrapped around an octamer, the 4 core histones, and there's 2 copies of each. So it's an octamer with a couple hundred bases of DNA wrapped around it. And there's 100 million of those that package up your genome into your nuclei of your somatic cells. Okay, so here's a whole nucleosome. There you go. You fly it by native MS. You get a mass to within a few Daltons. Good to go. Let's tickle it. Let's fragment it, and out pops the 4 core histones. So here's H4, H2A, H2B, and H3. And so the mind races. If you know chromatin biology, the mind sort of says, well, this is interesting. And then you could do that next step, like isolate histone H2B and fragment it. Here's all the fragment ions from H2B. And then you can start to match the fragment ions to the sequence as we've done for years in top-down. So I don't know if you want to call this top-down or over the top-down or sideways top, or who knows. The semantics are fun, but technologically I thought this was a cool experiment. And you can fragment each of the histones separately and in principle map their modifications, etc. And here's the first example of doing this, where we acetylate. We treat it with an acetyltransferase that takes acetyl-CoA and puts an acetyl group on lysines. And here they are, monoacetylated H3 ejected from the whole complex that was enzyme treated. And then 95% of that acetylation is sitting right at lysine 14, which is highlighted in red at the bottom part of this slide. Okay, so that's the first and second vignettes. I thought I would sneak in a third, and this is, I think, of high interest. And this, we're scheduled for 50 minutes. Is that right, Chris?
Yes. Yep. And then we'll switch to questions.
Cool. Okay. Yeah. Then we're right on target. And I don't know. I'm just talking into Zoom world and hoping that people are resonating, etc. But questions, definitely time for some questions at the end. Okay, so this is a change in the readout of ions. It's a fundamental shift in how to determine a mass spectrum. And it lives up more to that word, a mass spectrum, not an M over Z domain spectrum. It produces directly in the mass domain. And I guess, yeah, Phil and Jared have been absolutely critical to this. Fantastic colleagues, along with 20 other people over the years. And it comes from the veritable community, or the venerable groups of like Martin Gerald and Evan Williams. They've been working on this single ion world for a long time, showing in ion traps, which have frequency shifts for their different ions in these traps, how to read out spectra directly in the mass domain. And what they do is they assign the charge on a single ion in a trap. And oh, I pulled that slide for time's sake. But the trap is it's an anharmonic ion trap basically, and the single particle will move back and forth, and they'll estimate the charge to about 0.03 accuracy. They get very good charge state assignment in these traps. But they do one at a time, maybe 10 at a time. There was a paper last year. But let me show how we're doing it, which is really good frequency accuracy. Like an Orbitrap is locked down, good frequency accuracy in the Orbitrap. But the charge accuracy has always been the problem. It's like, okay, you would have a single ion in the ion trap spinning around. How many charges are on that? Well, I don't know. You plus or minus 5 charges. You couldn't do it. And that was the barrier, but we found a way to do it. And so let me show you the result, okay? And so if you mix 4 proteins together, and they're listed on the right-hand side of this slide, they're the usual suspects that you would buy from Sigma catalog. If you just mix them together, right, all their charge states from denaturing mode electrospray will overlap. And you get this charge state compression or congestion on your left-hand side in red. And now imagine that they weren't pure proteins, but they all had different proteoforms slightly. There was a mix of each of them. They each had their proteoform family. And now imagine you had 500 of them, okay? You would get what we have come to call the hump of death. You get no information. It's too complicated. Too many ions, too densely packed. But on the right-hand side, you can now get a mass spectrum, and I'll show you an example of that. And it's based on the Ryan readout. So because I'm amongst my kindred spirits here in mass spectrometry, let me just summarize it this way, which is that the readout of ions as an ensemble, let's say there's 1 million ions in each spectrum on the left-hand side, okay? So there's 1 million ions. uh, uh, in the trap, and you read them out as an ensemble, as a group, what's the basis for that readout? It is the constructive interference of the ions as packets, that as they move all around, you will see beat patterns. And if you've never studied this before in Fourier transform mass spectrometry, it's a good little homework, little quiz set to look at the time domain and say, oh, the reason I get signal is because of the constructive interference in the time domain. And I get these beat patterns and I get signal. Totally different on the right-hand side. On the right-hand side, you are monitoring the precession in an Orbitrap for each individual ion. And I'll show you how we do that. But the readout is completely different. Notice the baseline is completely different. And we do this using 1,000 ions at a shot now. Okay, and so I think there was some sense this was important. CNE News did a little write-up of it before the main article was published. And so how does this work? I think I need to skip over this due to time, but there's those anharmonic traps that's been used for a long time where you get frequency shifting, but really good charge state resolution. And now we're in an Orbitrap. And so here's one of these time domains. And these are the isotopic beat patterns. And it's from the constructive interference of the isotope peaks for a protein like myoglobin. But now we're going to, over a 2-second period, as the ions are precessing, we are going to integrate the induced current for a given ion over that 2 seconds. And the integrated current will provide a slope. And that is how we did it. The slope of that line is the charge. So if I have a 30+ charge state, I'm going to have a different slope, a higher slope, than a 29+ charge state, or a 28, or a 27. And that's how we know the charge state. We call these story plots, and they allow us to assign a charge to each and every ion. I should say about 90% of the ions survive long enough. And so here's a simple, these are 2 isotopic peaks of myoglobin, A and B. Here they are, they're the same charge state, it's the same isotopic cluster, and they have the same slope. So these are some early data, and I hope you have a sense for how it works. And the frequency position is just spot on. This is a single ion, and our ability in an Orbitrap to assign its frequency is very strong. That's the strong point. So strong, in fact, that you can get this ultra high resolution. You get 20-fold gain, 10 to 20-fold gain in resolving power because it's a single ion. You can nail the position. You know it's one ion. And so therefore, you know the position of it. It's the same. It's an analogous concept to like super resolution in imaging, in fluorescent imaging. If you have a single molecule fluorophore, you can locate its position with 10 to 20-fold more accuracy in space. And that's like storm type methods in microscopy. It's the analog of that in our world, mass spec. And so here's, we're getting 400,000 to into the 5-6 million range of resolving power, depending. And we have a paper out on this, and then another one coming up to, we've isotopically resolved up to 500 kilodaltons. And that paper is coming shortly. So that's the process. And I mentioned it was multiplexed. And let me do the time check. Okay. And this is all published. Sorry, this is an older deck, but this is now, this is last month's cover article in Nature Methods, April 2020. So you can go check it out there. It takes you through the whole process. And these are just the figures from the paper. And this is a supplemental figure. So the thing that was surprising to me was that either in native mode in blue on a UHMR, or the red in denatured mode in red on a Q-exactive plus, the calibration is stable. So what do I mean by calibration? It's the stability of this slope where, because from a story plot, the integrated current, we have to be able to go in the reverse way. We say here's an individual ion, here's its story plot result. What is its charge? We have to refer to this calibration plot. It's a calibration for story plots. So here's the story slope. And then we have to be able to go up and down and be able to assign the charge state to an integer value with confidence. And we can do that 96% confidently, or 96% of the time we get the right answer. So there's a little wiggle there, and we're improving that right now. Okay, so that's a sense of what we've done, and I won't take you through all the figures in detail, but this is the heavy and light chain. You know, if you compare left ensemble, standard MS to right charge detection, mass spectrometry, you know, you're just very different spectral attributes, very different operational considerations. The thing I'll mention is just high complexity. So in top-down, you know, I would be afraid of complexity, and now I'm not. Basically, that's the game changer for our lab, is that, you know, you bring me a couple hundred kilodalton target or a whole virus particle and say, I need a mass spectrum of this. I say, okay, we'll have it free tomorrow. And I can handle very dilute samples. So because you're dealing with individual ions, you dilute the sample. So instead of micromolar, you're down to low nanomolar as a routine, because you only can let in 1,000 ions at a time. So I think that's a big upside as well. On the complexity note, and I'll finish here and scoot through, and this is figure 2 from the paper, but this is that mixture of 500 proteoforms. It's actually more like about 1,000 masses, but we can assign 550 of them on the right because we don't have the hump of death, and we have a proteoform reference database, a proteoform atlas, and those mass values we can look up and get an identity from previous tandem MS studies. So, okay, I've talked for 50 minutes. There's GrowEL, it's in the paper. There's virus-like particles. So for those of you who are into AAVs and gene therapy with virus delivery, et cetera, it's important, as you imagine it would be, to look at the cargo. Anyway, I think I've spoken too long in a unidirectional way. I just want to thank Daniela, professor at Northwestern, and Ben, who made those VLPs possible. The team at Thermo here, that's led by Mike Sanko. Fantastic scientist is Mike. Those who know him, he's one of the great minds in mass spectrometry. It just happens to be hidden by his colleagues most of the time at Thermo. And all the funders of our lab, thank you. And I'll just move to thank the team, which developed I²MS, or individual ion MS, starting in 2017. And we've been running this project in high tilt mode, fast burn for a few years. And I just think it's a really important part of our future. So thanks for that. And I will just shut up, but please throw darts.
Thanks a lot, Neil. We really appreciate it. I'm going to open the floor for anyone that has questions. You can feel free to unmute yourselves and ask away. Nope, it looks like we've got one question here in the chat box regarding proteoform complexity. The question is, what about following proteoforms across life of the cell or during perturbation of the cell?
Yeah, so as proteins age, that's interesting. Like the proteins in your eyeball, they don't really turn over. But proteins in a liver cell that's active, metabolically active, they will turn over, most of them. And the rate of that turnover, I think we know a lot about that from bottom up. So if a protein is very fast turnover, it would be less susceptible to environmental accumulation of environmental xenobiotic type alkylation or environmental modification. That's less true of proteins in the blood. So basically, it depends. It depends on the biological context of the proteins and their half-life. Your eyeballs, I mean, if you look at a young person, look at your grandfather or grandmother, you very clearly see deterioration in the proteins of the eyeball. So following that in disease pathogenesis or neurology and proteinopathies as proteins age. I mean, it is the challenge of the century to really figure out how to interrupt some of these disease-related pathogenesis. And my point is, okay, so what are in those plaques in the brain? Exactly what proteoforms are there? It just seems like the forensics is lacking. And I think top-down is the state of readiness is such that we could really illuminate the proteoforms and how they progress through models of disease, mouse models, certainly neurological degeneration. And that's why I think we need a proteoform project to really just make that disruptively cheap, more democratized access to it, a revolution basically in technology. That's what we're up against. So I'll leave it there.
All right, looks like we have another question here in the chat box. Would ion suppression or proteoform suppression limit the capacity to detect the real complexity?
Yeah, so certainly. When you have, for example, where's that histone's a good example. So when you look at the history of histone H4 and the proteoforms, you start at the top. The most abundant proteoform is 50% of all nucleosomes have one proteoform, and it's N-terminally acetylated and lysine 20 dimethylate of histone H4. And then you descend into less and less abundant stuff, and the technology to get down there, how much is it encumbered by ionization suppression? I think that's the question. And I don't subscribe to that being one of the primary challenges. Like if you had a mass spectrometer that had a 10 to the 9 dynamic range in a single scan, if you could imagine such a beast, I don't believe that they're suppressed from ionization to the point where you wouldn't see them. So, I mean, I know that people throw out the word ionization suppression, particularly for native mode stuff, and they mean a variety of things. So it would take a while to disconnect all of the parts of that. But I don't believe that one dominant proteoform would suppress others from detectability by a log or 2 logs, like a massive suppression effect. I don't subscribe to that.
All right. Is there a significant mass bias when using charge detection mass spec? And do you envision this technique being used for quantitative mass spec?
I do. I think there's actually less bias. The mass bias, there is a bias, and I'm searching desperately for a slide. Okay, so we just published in JPR a fragment ion readout. So a top-down, here, let me see if I have this. If I'm really on my game, I have these reserved slides. Basically, the punchline is there is more bias for higher mass. The higher mass you go, the better the charge detection mass spec gets. And I don't have that slide, sorry. But in JPR, I guess it was 2 months ago, we fragmented a 40 kilodalton protein, and we looked at the fragment ions by charge detection and I squared MS. And typically, for ensemble measurements, there's a really strong low mass bias. We see the B and the Y ions below position like 60, the B 60 and the Y 60 and below. That's our strong bias for reasons. But for I squared MS, we see the big ions. We see the complementary pairs like the B300 and the Y300. It's a really flips it upside down. So I think the bias is just, and I'll say on the flip side, that's the good side, high mass, no problem. Out to megadaltons, you just get this mass readout. That's it. And really flat bias across the way. But in the range for that we're looking at for fragment ions, we can, or MS1 intact ions, we can only go down to 6+. in charge detection. We cannot assign charges below 6+. The signals, the story plots, it's just a non-starter. Oh, now I can see the chat. Yeah. Okay.
So the next question that we have here is, given that many extracellular proteins are heterogeneously glycosylated, how much more complicated does glycosylation make things for top-down?
Yeah, if you said just intact mass, I'm like, hey, I can have that for you by Tuesday. Seriously, just if you want the glycoproteoform readout, the pattern, done. And on dilute samples, I'm not overselling this advance. But for down, you want to put the down, you want to start fragmenting, start localizing N-glycosylation sites, especially if you get to 4, 5, 6 N-glycosylation sites that are high stoichiometry, complicated glycoform patterns. That's not for the faint of heart. But for just patterns, MS1, you know, it's like, you know, no problem. We'll make our 3:30 tea time.
All right. Does anyone else have any other questions? You don't have to type them in the chat box. You can certainly unmute yourself and ask if you'd like.
I think it's 5 o'clock, so I'm allowed to have a small nip here.
Looks like we have one more question for you there. You dominantly showed HCD, quad-style fragmentation. What's your opinion of UVPD or ETD?
Yeah, so when you really want to localize sequence variation or PTMs, those come in really handy for that. And I'm excited to combine those two types of fragmentation methods with I²MS because I just think the density and of the information will be incredible. So we typically roll in high throughput mode with HCD. Native, we are doing native top down with HCD by far, you know, predominantly. We just got native mode, you know, in the past year or so, working on the Eclipse. So the Eclipse is capable of going out to 8,000. And so you know, you can start to pursue those things. We haven't, just for bandwidth issues. But we did have a high throughput UVPD discovery mode top down paper in MCP last year. But yeah, if you have a targeted case where you also can sum a lot of fragmentation scans or go fast with the Eclipse, it depends on how much you got to localize and how much time, whether it's a chromatographic time scale or not. But yeah, for antibodies, I think I do have that, and I know we're in bonus time here, but I do have the antibody, like for MABs and ADCs. we published a paper in MABs that showed doing all the types of fragmentation, you know, basically 50–70% sequence information for heavy and light chains. Yeah, using all those fragmentation techniques.
Well, if there are no more questions, Neil, I'd like to thank you, and for all of the muted attendees. Nope, it looks like there's one more question that came in. Can top-down be, well, I think he may have already answered that question, but can top-down be used quantitatively?
Oh, for Steve, you bet. Yeah, so within a proteoform family, if I have 5 proteoform families, 5 proteoforms in a family, that information's quantitative, right? The relative ratio, that gives you, and if one or another shrinks or changes in state A, state B, that is really biologically, on many cases we've shown are really valuable and reliable. It's like their own internally calibrated group, and you can detect biology changes. So in that case, yes. And then we do this global quant, like 0 to 30 kilodaltons for biomarker discovery. We've got that down. We've been publishing on that for a long time. That's our biomarker discovery platform, is the 0 to 30 label-free quant. There's no TMT or iTrack, sorry, for top-down, but we just use good study design, randomized sample order, you know, normalization, label-free, top-down, and you get out your volcano plots and your discovery data sets. So those are two different contexts in which you can quantify.
All right, wonderful. Well, Neil, again, thank you very much. One last question, or one last opening to anyone else that has any more questions. Does anyone else have any other questions for Neil? I know we're running a little bit over, but if anyone has anything else.
Thanks to Nick D there. Yeah, right on, Nick. Good luck in your career. Oh, that was privately. Sorry, dude. Anyway, thanks, Nick. No problem.
Neil, we really appreciate it. And again, from all the attendees here that are all muted, and I'm sure they would say the same thing, thank you very much. And one last note before everyone else jumps offline. This was really more of a note to the Delaware Valley Mass Spec discussion group participants, but please fill out the annual survey that we sent around. We're going to send out another email afterwards. But Neil, we thank you very much and appreciate your time. Everybody, you stay safe and stay healthy.
Yep. Thanks, Neil.
Yeah, thanks, Steve. Kevin.